Hello and welcome to Chapter 7. The article at the beginning of Chapter 7 is concerning actually a heating cooling unit that's part of the lung heart machine that's used in open heart surgery. And we'll talk a little bit about this later on, of how can we make sure that all of the equipment that people are subjected to in the hospital actually are going to be clean enough from a bacterial or a microbial standpoint. So we'll talk about that a little bit later on. In this chapter, we're going to talk about nutrition for the microbes. And so the first question that we really need to ask is, what is nutrition? Well, nutrients are acquired from the environment and are used for cellular activities. And this is true of all nutrition for all species. An essential nutrient is any substance, whether in elemental or molecular form, that must be provided by the environment to the organism. In other words, the organism cannot make it on its own. Macronutrients are those nutrients that are required in relatively large quantities by the organism, and they usually play principal roles in cell structure and metabolism. Micronutrients or trace elements are present in much smaller amounts, and in fact some, called the ultra-trace elements, are needed in only very tiny amounts. Most of these are involved in the function of enzymes and the maintenance of protein structure. Now, we can divide nutrients into the inorganic nutrients and the organic nutrients. Inorganic nutrients are usually simple molecules that contain a combination of atoms other than carbon and hydrogen. So for example, H2O. It does contain hydrogen but not carbon, therefore it is classified as an inorganic nutrient, and all cells needs plenty of water. Organic nutrients contain both carbon atoms and hydrogen atoms. Usually these are the product of living things, and we'll talk about the organic material here in just a moment. So if we were to look at this table and look at the principal elements from the periodic table that are needed for living organisms, we certainly can see that carbon, because all of life as we know it on Earth is a carbon-based life form. Certainly we need oxygen, we need nitrogen, hydrogen, phosphorus, sulfur, potassium, sodium, calcium, magnesium, chloride. All of these are primarily macronutrients. And then in some, we need iron. In humans, iron is considered to be a macronutrient, while in other organisms, it's a micronutrient. And then these others actually down here that are have various functions in the body are actually usually micronutrients. If we were to analyze the chemical composition of a typical E. coli cell, proteins make up the bulk of the weight by total weight, and almost 50% of the actual dry weight. And what atoms do we see in proteins? Well, we have carbon, oxygen, hydrogen, and nitrogen, and that's the definition of the proteins. We'll have nucleic acids, both RNA and DNA. RNA is about 20% of the dry weight, while DNA is about 3% of the dry weight. So it's about a 6-to-1 ratio of RNA to DNA. Certainly we need carbohydrates because they are our primary energy source to produce ATP in the cells. We need lipids as they are the secondary energy source. And then there'll be a variety of miscellaneous things. That's of the organic compounds. Of the inorganic compounds, 70% of the total weight of an E. coli cell is water. That's also true of a human being. About 70% of our total body weight is actually water. And then all the other inorganic compounds, things like sodium, potassium, magnesium, calcium, all of these usually are ions. They make up about another 1% of the total weight. So, where do we get our carbon? If you are classified as a heterotroph, this is an organism that must obtain its carbon in some form of organic chemical, such as carbohydrates or lipids. You are usually dependent, therefore, on other life forms for your carbon source. Most carbon sources exist in the form that is simple enough for absorption, so the simple sugars, the amino acids. And many carbon sources must be digested by the cell in order to be absorbed. If you are an autotroph, or self-feeder is literally what the word means, this is an organism that can use inorganic CO2 as its carbon source, so carbon dioxide. These have the capacity to convert carbon dioxide into carbon compounds within the cell that they then can use for energy production. Typically, autotrophs are not dependent on other living things, but they are usually dependent upon photosynthesis. So where do we get nitrogen? Well, the main reservoir of nitrogen is actually the nitrogen gas that we find in atmospheric air. It actually makes up approximately 79% of the atmospheric air surrounding us. Nitrogen is an indispensable component for DNA, RNA, ATP, and other protein structures. The primary nitrogen source for heterotrophs usually must be degraded into basic building blocks in order to be utilized. So primarily for heterotrophs, this is going to be our protein source. Some bacteria and algae, however, can utilize inorganic nitrogenous nutrients, such as nitrate, nitrite, or even ammonia. Regardless of the source of nitrogen, nitrogen must be converted to ammonia before it enters the cell. This is the only form of nitrogen that can be directly combined with carbon in order to synthesize amino acids and other compounds. Oxygen plays a very important role in the structural and enzymatic functions of the cells. It's a major component of the four major organic substances: carbohydrates, lipids, proteins, and nucleic acids. It's also a common component of inorganic salts. Oxygen makes up approximately 21% of the atmospheric air. Hydrogen sources. Hydrogen has overlapping roles in the biochemistry of cells. First of all, it's very important in maintaining pH. It also forms hydrogen bonds between molecules in order to form many of our large macromolecules. And it serves as a source of free energy in the oxidation reduction reaction of aerobic respiration. Phosphorus, the main inorganic source, is phosphate. It's derived from phosphoric acid, and in fact the phosphate ion is actually an ionic form of phosphoric acid. Phosphate is normally found in rocks and in oceanic mineral deposits. Phosphorus is a key component in the nucleic acids. It makes up that sugar phosphate backbone, and it's essential to the genetics of all cells and viruses. It's found in ATP, adenosine triphosphate, which is the only form of energy molecule the cell can actually use to do work. Sulfur is widely distributed throughout the environment in rocks and sediments. It's an essential component of many vitamins, such as vitamin B1, and amino acids, such as methionine and cysteine. There are several of the amino acids that contain sulfur, as well as the carbon, oxygen, hydrogen, and nitrogen. Sulfur forms disulfide bridges that help determine the shape and structural stability of many proteins, including our antibodies in the body. Other essential organic nutrients will include certain growth factors. And these growth factors actually are usually an organic compound, such as an amino acid, nitrogenous base, or vitamin that cannot be synthesized by the organism. An example, the essential amino acids in human. Of the 20 amino acids that we use to build our protein, 11 of them are essential. We must get them from the environment. How do microbes actually feed? Where do they get their source of energy? Well, if you're a phototroph, these are microbes or cells that can use photosynthesis. So they're going to convert light energy into chemical energy. Chemotrophs, on the other hand, are microbes that gain energy from chemical compounds, and these can either be inorganic sources or organic sources. So we can utilize classification of how cells actually get their energy and their carbon source. So I said earlier, if you're an autotroph, you're going to use CO2 as your carbon source. If you use sunlight as your energy source, then we can actually add photo onto the name. And so a photoautotroph, the carbon source is CO2, the energy source is sunlight, and this would include photosynthetic organisms such as algae, plants, and the cyanobacteria. If you get your carbon from CO2, but your energy source is simple inorganic or organic chemicals, then we'll say that you are a chemoautotroph. And only certain bacteria or archaea, such as the methanogens and deep sea vent bacteria, actually are chemoautotrophs. And since these usually don't cause any problems in humans, we really won't talk about them. Now, if you get your carbon from an organic substance, such as carbohydrates, but you use sunlight as your energy source, then we're going to call you a photoheterotroph. And this would include the purple and green photosynthetic bacteria, which again don't cause disease in humans, so we're not going to talk about them. However, if you use an organic compound for your carbon source, and you use organic material for your energy, then we're going to refer to you as a chemoheterotroph. And this would include many of our fungi and bacteria, but we can also break down the chemoheterotrophs into this. If you utilize living tissues as your source of your organic compounds for energy, we call you a parasite. If you metabolize the organic matter of dead organisms, then you're a saprophobe. So photoautotrophs capture energy from light rays and transform it into chemical energy that can be used for cell metabolism, and their carbon source is CO2. Through the process of photosynthesis, they can produce the organic molecules that can be used by themselves and even heterotrophs. Plants fall into photoautotrophs, and think about it. Plants actually make their own glucose from using CO2 and light to produce the glucose, and then they can use that glucose to produce ATP. But we eat the plants, we can then use the glucose the plants made in order to use to make that glucose, or to use that glucose to make our ATP. And so photoautotrophs usually form the basis of most food webs. Chemoautotrophs, chemoorganic autotrophs, so they get their CO2 or get carbon from CO2, but they use some sort of inorganic or organic source of energy. Most of them do use organic compounds for energy and an inorganic compound as a carbon source. Chemolithoautotrophs require neither sunlight nor organic nutrients and totally rely on inorganic materials completely to make their energy. The methanogens. These are chemoautotrophs that produce methane from hydrogen gas and CO2. They're formed in anaerobic hydrogen-containing microenvironments. For example, in the ocean, at a particular depth, there is actually a layer of methane gas, and that's where many of these methanogens actually live. They oftentimes will live also in the deep ocean vents and hot springs. Methane that's produced by these methanogens can actually be captured and used as a fuel, but this may play a role as a greenhouse gas. Chemoheterotrophs, the majority of heterotrophic microorganisms are going to be chemoheterotrophs. They derive both their carbon and energy from organic molecules. So, for example, our cells, where do we get our ATP from? We get it from glucose. Where do we get our carbon from? We get it from glucose. So organic molecules are processed through respiration or fermentation and produce ATP, giving off as a byproduct CO2. Aerobic respiration, as the name says, it does require oxygen. And this is the principal energy-yielding pathway in animals, protozoa, fungi, and the aerobic bacteria. Glucose and oxygen are reactants, and carbon dioxide and water are given off as byproducts. Earth's balance of energy and metabolic gases is actually dependent upon aerobic respiration. It is complementary to photosynthesis, and in fact what we'll find later on is that photosynthesis is the exact opposite chemical reaction to aerobic respiration. Saprophobes are free-living microorganisms that feed primarily on organic dead matter. These are going to be the ones that, like fungi and bacteria, that are going to be our decomposers in the environment. While parasites derive their nutrients from the cells or living tissues of a host. So the saprophobes are big decomposers, and they decompose plant, animal matter, dead microbes. They are very important in our environment in the fact that they recycle the organic materials back into the soil or into the environment in some way. Most saprophobes have a rigid cell wall and cannot engulf larger particles of food, and so they have distinct ways of actually bringing the nutrients into the body of the cells. They release enzymes into the environment that can digest the food into small particles that then can be transported through the cell membrane. So this is just showing, here's some organic debris, and this cell wall is really a barrier to these large molecules. So the organism will produce digestive enzymes that are transported to the outside that will actually break down those organic debris into very much smaller particles, which then can be transported across the cell wall and the cell membrane into the cytoplasm of the cell. Parasitic microorganisms that live in or on the body usually cause some degree of host or harm to the host. And so they are considered pathogens because they can cause tissue damage and can even cause death. Ectoparasites live on the body, while endoparasites live in organs and tissues. Intracellular parasites actually live within a cell, and an obligate parasite is unable to live outside or off of the living host. Now, necessary nutrients must be taken into the cell, and waste materials have to be transported out of the cell. And so we need to talk a little bit about transport across the cell membrane. Remember that the cell membrane is a selectively permeable membrane, and so there have to be structures that are specialized for transport. The cell wall is actually pretty non-selective to screen for the entrance or exits of molecules other than simply based upon size. So let's review the movements across a cell membrane. Diffusion. This is the movement of molecules in a gradient from an area of higher concentration gradient to an area of lower concentration gradient. Diffusion across a cell membrane is determined by the concentration gradient and the permeability of the substance. We have both simple diffusion, which is simply moving from an area of high concentration to low concentration with no barriers, and facilitated diffusion, where we can't just go anywhere through the membrane, but we have selected pores or proteins that we can use as transports through the membrane. So, good example of simple diffusion. What if you dropped your sugar cube in your coffee? Well, to begin with, the sugar is in high concentration right here and low concentration out here. Same thing if you drop a piece of potassium permanganate, that's purple stuff, into a beaker of water. To begin with, we have a high concentration right there. But over time, what will happen is all of the potassium permanganate or sugar molecules will equally disperse throughout the solution. And so no matter where I chose, I would find the exact same number of the solute, what's being dissolved, in the solution. And this is known as reaching equilibrium. And diffusion will continue until equilibrium can be reached. Osmosis is the movement of water across the selectively permeable membrane. The membrane is selectively or differentially permeable, but it has passageways that allow for the passage of water, but not other substances. And what we find is if we take a sugar solution, let's say, and put it into a dialysis bag here, I can tell you that the dialysis tubing will allow water to move through, but not the solute, whatever this is. And so water can move through the bag, but the solute can't. Over time, what's going to happen is that water, being in high concentration out here in the beaker and less concentration in the bag, that the water is actually going to move into the bag to try to equalize the amount of water on both sides. And what this will do is actually increase the volume within the bag, and since this is attached to a tube, the volume will actually go up. So what we can say is that osmosis is the movement of water from high water concentration to an area of lower water concentration. But why is there less water in the bag? Is because we have dissolved solutes in here. So we have lots of solutes in the bag, but very few solutes out. So water is going to move into the bag where there's a higher degree of solutes to try to equalize the amount of water on both sides. If both solutions on either side of a cell membrane or membrane are identical, we refer to that as an isotonic condition. So the solute concentration in the internal and external environment are exactly equal, therefore water would be equal, and therefore we get no net movement across the membrane. So diffusion of water proceeds at the same rate on both sides. So if a molecule of water goes out of the cell, one has to move in order to maintain equilibrium. Isotonic solutions are generally the most stable environment for cells because there's already an osmotic steady state within the cell. Hypotonic conditions means that the solute concentration of the external environment is lower than that of the cell's internal environment. Therefore, we're going to have more water outside the cell than inside the cell. Pure water is the most hypotonic environment because it has no dissolved solutes in it. And the net direction of the movement of water is from the hypotonic solution outside into the cell inside. Cells without walls then will swell and rupture because equilibrium will be very hard to get to before the cell membrane will actually break. Hypertonic conditions are the exact opposite. Now the environment outside the cell has a slightly higher concentration of solutes than inside. Therefore, outside the cell, we have less water. The high osmotic pressure forces water to diffuse out of the cell, and the cell will actually lose water and shrink. Hypertonic conditions will limit the growth of microbes, and I'll explain that here in just a moment. And this is really the principle behind using concentrated salt or sugar solutions to preserve food. We are creating a hypertonic environment outside the microbial cells, and they're going to lose water and not be able to reproduce. So in an isotonic solution, both a prokaryotic and eukaryotic cell without the cell membrane are going to be just fine because, again, water is already equal on both sides because the solute concentration is equal on both sides. So no net movement. For every molecule of water that moves out, one's going to move back in. That will maintain that equilibrium. However, if we put both types of cells into hypotonic solutions, so now there's less solute on the outside, therefore more water, and water is going to want to move into the cell. In eukaryotic cells without the cell membrane, well, what's going to happen is as water moves into the cell is going to swell, and again, before we can probably reach equilibrium of the water inside and out, the cell membrane is probably going to rupture, and we get lysis of the cell. However, the bacteria, because of that strong cell wall, actually resist lysing, and so this will actually keep this cell from being destroyed in this hypotonic solution. But if we were to put these cells in a hypertonic solution, the eukaryotic cell will lose water and shrink, and it actually forms these kind of little wrinkles known as crenation. But here's what happens to the bacterial cell. This cell is going to lose water and shrink. The problem is the cell wall is rigid. The cell membrane is not. It will shrink. And as it is pulled away from the cell wall, it will actually be disrupted and will actually leak contents out of the cell, and the cell will actually die. And this process is known as plasmolysis. So the cell doesn't burst and rupture, but it actually is shrinking and tearing the cell membrane, and that causes death of the cell. So eukaryotic cells are much better able to withstand hypertonic solutions, while prokaryotic cells with their steady cell wall are much easier to survive in hypotonic solutions. Now, since isotonic conditions pose little stress to the cell, everybody likes it isotonic. But is the world a perfect place? Well, it's not. So what happens if these cells are put into hypotonic environments? Well, so for example, bacteria and amoeba living in fresh pond water, that fresh water would be hypotonic to these bacteria. The bacteria don't mind so much because the cell wall protects the cell from actually bursting and lysing. The amoeba do not have a cell wall, so how do they keep themselves from bursting? Well, they actually utilize a special organelle known as a contractile vacuole that basically constantly removes excess water out of the cell. This does require ATP energy, but it basically bails out the water as fast as it comes in, and so the amoeba can survive. In hypertonic environments, cells must restrict the loss of water to the environment or increase the salinity of the internal environment. And halophile bacteria are really good at this. They actually absorb the salt from the environment to equalize solute concentrations on both sides, then making the cells isotonic to the environment. And so these halobacteria actually have a physiological need for high salt concentration in their environment in order to maintain an isotonic environment inside. Facilitated diffusion is really a mediated transport. We're still going from low to high concentration, but we utilize a carrier protein that will bind a specific substance and carry it across the membrane. Carrier proteins usually exhibit specificity, meaning that these carrier proteins will only bind and transport one or a few types of molecules. But facilitated diffusion does not require any energy expenditure. The binding actually changes the conformation of the carrier molecule so that the substance is moved across the membrane, and that requires no energy. So, just a little review. Simple diffusion, going from an area of high concentration to low concentration. Facilitated diffusion, same thing, except we go from a high concentration to low through a carrier molecule, expending no energy. Now, a term that you should be familiar with is saturation. This is the rate of transport of a substance that is limited by the number of binding sites on the transport proteins. The rate of transport increases as the rate of substance concentration increases until all the binding sites are occupied. So if I were to put in 30 grams of sodium chloride into 100 mils of water and stir it all up, the salt would actually dissolve or diffuse throughout the solution. However, if I were to put in 36 grams of sodium chloride in 100 milliliters, the solution can only hold 30 grams, and so the additional 4 grams actually will remain undissolved. And it wouldn't matter if I put in 50, it would still only 30 grams would actually dissolve within the water because basically that's all it can hold. Same would be true for any binding sites on molecules that are moving things across the membrane. If all the binding sites are busy, I can only move that much, and that's it. I can't go any higher. Now, we can then see competition, and this is when two molecules of the same shape bind to the same binding site on a carrier protein. The chemical with the higher binding affinity, or the chemical in higher concentration, will be transported at a greater rate. So if I have simply chemical A and chemical B that are both competing for a carrier, if I've got twice as much A as B, A is going to be transported more frequently than B will be. If the carrier protein, however, likes B better, and I have equal numbers of A and B, B will actually be transported at a higher rate than A because of the higher binding affinity. Now, active transport is going to require energy, and this transports nutrients against a concentration gradient. Or, in other words, we're going from low concentration to high concentration. The presence of specific membrane proteins are necessary. We are going to need a transport of some sort, and this can include permeases, which are specific enzymes, or protein pump systems. And we're going to have to use ATP in moving these particles. Think about it. Water wants to go which way on the hill? The water wants to run down the hill from an area of high to low. That's what we saw with our passive movements of diffusion and facilitated diffusion. But now, can you take water up a hill? Well, you can, but you're going to have a carrier, and you're going to have an expenditure of energy. Specialized pumps such as the potassium, sodium pump, or the hydrogen pump will actually move these molecules against their concentration gradient using a carrier protein and ATP. Now, there's also another type of movement that you're probably not as familiar with, and this is group translocation. This actually couples the transport of a nutrient with a chemical conversion to another substance that is immediately useful inside the cell. Endocytosis is going to be used for the transport of large molecules or particles or liquids across the cell membrane, especially by certain eukaryotic cells. This does require the expenditure of energy in order to move the cytoplasm and the cell membrane. And you've heard the terms phagocytosis, and this is just endocytosis by cells that ingest whole cells or large solid matter, and they bring them inside in what's known as a phagosome. Pinocytosis simply means cell drinking, and this is the entry of oils or liquid molecules into the cell. However, pretty much pinocytosis has fallen out of the vernacular because, again, we don't say you sit down and eat and drink. We know that you're going to be taking in some water with the solid particles as well. So active transport, this is going to be using energy and a carrier molecule transporting something from an area of low concentration to high concentration. Group translocation, we're going to move a particle through, but as we do so, we're going to chemically change it using ATP. And this is a very important step in the energy production because this is actually going to be the first step of glycolysis, and we'll talk about that in glycolysis. And then bulk transport using phagocytosis or pinocytosis, simply again just bringing those larger particles into the cell in a phagosome. So, can you compare and contrast macronutrients and micronutrients? A what requires organic molecules for both its carbon source and energy source. True or false? Water requires a specialized membrane protein in order to enter or exit a cell. And what type of environment has a higher concentration of solutes outside the cell than inside the cell? Environmental factors that will influence microbes. Well, certainly the temperature, so heat, cold. What gases are available? How acidic or alkaline is the environment? Is there any radiation that could damage DNA or RNA? What is the osmotic pressure inside and outside the cell? What is the hydrostatic pressure? And then what about other microbes? Well, temperature. Let's start with that first. The cardinal temperatures is a range of temperatures for a given microbial species. The minimum temperature and maximum temperature, these are the extremes. The minimum temperature is the lowest temperature that a microbe can survive, while the maximum is the highest. The optimum temperature is where we get the best growth of the microbe. So the lowest temperature that permits a microbe's continued growth and metabolism. While it's not going to be great growth, that at least will survive. Below this temperature, the activities of the microbe are limited. In fact, usually the microbe will actually die. Maximum temperature, the highest temperature at which growth and metabolism can proceed. If the temperature rises slightly above the maximum, typically growth will stop and the organism will die. And actually, I'll say this, heat kills faster than cold. If the temperature continues to rise, the enzymes and nucleic acids will become denatured or completely unravel and become permanently inactivated, and the cell will actually die. So what we like is the optimum temperature, and this is the intermediate temperature range between its minimum and maximum. This promotes the fastest rate of growth and reproduction and metabolism in the microbe. Small chemical differences in bacterial membranes, which affect their fluidity, allow them to thrive at different temperatures. So let's take a look at this temperature graph. So you'll notice here in the green, we'll start with this guy first because these are known as mesophiles. Meso means middle, so kind of in the middle of this extreme range of temperatures. And you'll notice the minimum is down here at about 13 degrees Celsius, and its maximum is at about 47 degrees Celsius. The optimum temperature is at 37 degrees Celsius, which is body temperature. Now, psychrophiles, and phile means to love, and so psychro referring to cold. The psychrophiles actually have the temperature range here from about minus 20 degrees Celsius to about 15 degrees Celsius, but their optimum growth temperature is at about 4 to 5 degrees Celsius. That's basically just above the freezing mark, and so basically refrigerator temperature. Then we have a group that's kind of squished in between, the true psychrophiles and the mesophiles, and that's known as the psychrotrophs. So they're cold, moderated organisms, and their maximum growth or optimum growth temperature is at about 25 degrees Celsius, which is about room temperature. The thermophiles, way up here, usually at about 70 degrees Celsius is their optimum temperature, but they can survive down as low as about 45 degrees. And then the extreme hypothermophiles, they can get out here to 125 to 130 degrees Celsius, and it depends on whether we're talking bacteria or archaea. Some of the archaea go out even further here. So, why am I not really worried about the thermophiles or extreme thermophiles causing disease, or even the psychrophiles? Well, because basically they can't live at 37 degrees Celsius. So what group of microbes am I most concerned about with the temperature range? That's the mesophiles. So psychrophiles, again, typically have an optimum temperature below 15 degrees Celsius, and they can survive even in polar ice caps. So they are capable of growth at the freezing mark at 0 degrees. They typically will not grow, in fact actually are completely killed by temperatures above 20 degrees Celsius. The psychrotrophs or facultative psychrophiles grow slowly in the cold and have that optimum temperature right around room temperature at 25 degrees. Mesophiles, the majority of the medically significant organisms, because individual species grow anywhere from about 10 to 50 degrees Celsius, but the optimum growth temperature is body temperature at 37 degrees. So most human pathogens like it right at 37. And so why would we maybe not want to take the fever down of an individual? Let you think about that for a little while. Thermoduric microbes survive short exposures to high temperatures, and these can be common contaminants of heated or pasteurized foods. And the thermophiles generally have optimal temperatures above 45 degrees. They typically live in soil and water associated with volcanic activity or compost piles. The average temperature of the average compost pile is about 55 degrees Celsius, and habitats that are directly exposed to the sun. Extreme thermophiles, again, they grow between about 80 to 121 degrees Celsius. So why is our autoclave set to run at 121 degrees Celsius? I'll let you think about that question as well. Now, in relation to gases, microbes fall into one of three categories. Those that use oxygen and can detoxify the abnormal oxygens, those that can neither use oxygen nor detoxify it, and those that do not use oxygen but can detoxify it. Well, what am I talking about with detoxification? As oxygen enters into cellular reactions, some of it can be transformed into several different toxic products. Singlet oxygen, just a single oxygen or an oxygen molecule that has lost some electrons or a proton. These actually can cause damage to DNA and RNA. They are extremely reactive molecules produced by both living and non-living processes. They oftentimes are produced by phagocytes to kill invading microbes. But buildup of singlet oxygen and the oxidation of the membrane lipids and other molecules can actually damage or destroy the cell. These are actually sometimes known as free radicals. Superoxide oxygen or superoxide ion oxygen, which has gained an electron, or hydrogen peroxide or hydroxyl radicals. All of these also are free radicals and can be destructive metabolic byproducts when we use oxygen in metabolism. Cells use enzymes typically to scavenge these and to neutralize them, such as catalase and superoxide dismutase. So, how do microbes process oxygen? Well, if an organism is an aerobic organism, that means it can use gaseous oxygen and in fact actually usually has to have gaseous oxygen in its metabolism. It possesses the enzymes needed to process toxic oxygen products that are produced. And so an obligate aerobe is any organism that cannot grow without oxygen being present. Facultative anaerobes, however, are aerobic organisms that do not require oxygen for its metabolism. While it prefers oxygen, it is capable of growth in the absence of oxygen. It can metabolize energy by aerobic respiration when O2 is present, but will switch and adopt to anaerobic metabolism of some form when oxygen is absent in the environment. Microaerophiles, micro, small, air, little, these do not grow at normal atmospheric conditions of the 21% oxygen. They require only about a smaller amount of oxygen, about 10 to 12% in the environment for their metabolism. So they usually live in a habitat that provides a small amount of oxygen but is not directly exposed to the atmosphere. So again, I'm not too concerned about these, except maybe getting into certain areas where oxygen is a little bit lower, such as the gut. An obligate anaerobe lacks the metabolic enzyme systems for using oxygen in energy production. And strict or obligate anaerobes cannot tolerate free oxygen and actually will die in its presence. They typically live in highly reduced habitats such as in lake sediments, oceans, even soil. Aerotolerant anaerobes tolerate oxygen. They do not utilize oxygen at all, period. They don't care if oxygen is present or not, they don't use it. They do complete anaerobic respiration. So they can survive and grow in a limited extent in its presence. They really don't care if oxygen is present or not. So they're not harmed by oxygen because they possess alternative mechanisms for breaking down peroxides and the superoxides that may be produced in the environment. So let's look at where these microbes would grow best in a solid deep tube. So if you're an obligate aerobe, where are you going to grow? Well, you're going to grow right at the surface because that's where most of the oxygen in the tube is going to be. Oxygen will diffuse down through the solid media, but basically by the time you get down here to the bottom of the tube, there's very little oxygen, if any. And so they just simply can't grow down there because there's not enough oxygen. If you're a facultative anaerobe, again, I said these guys prefer oxygen. So you're going to see the best growth right up here at the top because that's where the oxygen is. But they'll grow throughout the rest of the media because they switch over to anaerobic respiration. And so they can grow down here where there's very little oxygen as well. Obligate anaerobes don't want anything to do with the oxygen at the top, so they're going to grow right at the bottom of the tube. The aerotolerant anaerobes pretty well grow throughout the entire media equally. There's no best growth anywhere because, again, they don't care if oxygen is present or not. They're going to be doing aerobic respiration only, and so they just simply don't care where the oxygen is. And then the microaerophiles are going to pretty much grow right in the middle of the tube where there's about 10 to 12% oxygen. And so they don't like it with none, and they don't like it with atmospheric oxygen, but they like it kind of in between. The capnophiles, these grow best at higher CO2 concentrations than's normally present in the atmosphere. And so to grow capnophiles, we actually have to either get rid of the oxygen completely, and we need to add in CO2 in the environment. And this is important in the isolation of some pathogenic organisms. We actually have in our laboratory a CO2 incubator that will take oxygen out of the atmosphere and put CO2 in. So incubation is carried out for these microbes in a CO2 incubator that provides about 3 to 10% CO2. Normally, CO2 in the atmospheric air is about 0.3 to 0.4% of the total atmospheric air. pH. pH is that degree of acidity or alkalinity or basicity of a solution, again expressed on that scale from 0 to 14, with pure water being neutral at the pH of 7. As pH value decreases towards 0, the acidity increases, and as pH values increase toward 14, alkalinity increases. And so some microbes like to grow at certain pHs. And just like we saw with our temperatures, they typically tend to have a optimum pH. So acidophiles, the acid-loving ones, these require an acidic environment for growth. And most acidophiles have an optimum pH of about a pH of 3. There are many molds and yeast that tolerate acids and therefore are common spoilage agents even of pickled foods that are highly acidic. Alkalinophiles typically live in those areas that are hot pools or soils that contain high levels of basic minerals. And bacteria that decompose urine actually create alkaline conditions. And then the neurophiles grow best right around a pH of 7. And so if I'm looking at pathogenic microorganisms, I'm going to be primarily looking at the neurophiles because our pH in our body primarily is right around 7.35 to 7.45. Osmophiles, these live in habitats with high solute concentration, and we've already talked about the obligate halophiles that require high concentrations of sodium chloride actually for their growth. These have significant modifications to their cell walls and membranes and will lyse very easily or undergo that plasmolysis in hypotonic environments. Facultative halophiles can be resistant to salt, even though they normally do not reside in these high salt environments. Barophiles, these can withstand high hydrostatic pressure. And so think about the deep sea microbes that exist in pressures up to 1,000 times atmospheric pressure. By the time you go down in the Marianas Trench, which is 7 miles down, the pressure is extremely high. And so these barophiles are so strictly adapted to these high pressures that if you brought them to the surface without being pressurized, they would rupture when exposed to normal atmospheric pressures. Now, radiation. Phototropes can use visible light rays as energy source, but they can use protective measures against the UV radiation, such as yellow carotenoid pigments will absorb and dismantle toxic oxygens, and other microbes use enzyme to overcome the damaging effects of UV radiation on their DNA. We also need to talk about the association between organisms. And organisms live either in symbiotic relationships or non-symbiotic relationships. And all organisms live in some sort of community. No man is an island, the old saying goes. Symbiotic relationships are where organisms live in very close nutritional relationships, and usually the relationship is required by one or both members. Non-symbiotic organisms are free-living. They could live on their own, but oftentimes there's strength in numbers, and while the relationships are not required for survival, they may be actually advantageous. So let's look at the non-symbiotic relationships first. If the members cooperate and share nutrients between the organisms, this is known as synergism. And again, while the organisms could live on their own, they are better together because they cooperate and share nutrients. However, in some scenarios, non-symbiotic relationships are actually antagonistic. This is where some members will be inhibited or destroyed by others. Actually, our normal microbiome in and on our body works with this antagonism. The normal microbiome is actually antagonistic towards other microbes moving into their territory, and so will actually destroy them. Symbiotic relationships can be mutualistic, commensalistic, or parasitic. Mutualism is where both organisms benefit from the relationship, and usually this is an obligatory dependent relationship. They must live together in order for both of them to survive. Commensalism is one member of the group benefits while other members will not be harmed, but they neither benefit nor are they going to be harmed in any way. Whereas parasitism is the parasite is dependent upon the other in the relationship and will benefit from it, while the other member actually will be harmed. So symbiosis is a general term used to denote a situation in which two organisms live together in a close, usually nutritional partnership. Mutualism exists when organisms live in an obligatory but mutually beneficial relationship to both parties. Commensalism, one organism receives benefits while the other one is neither harmed nor benefited. So the commensal organism receives the benefits, the co-inhabitant is neither harmed nor benefits. Satelliteism arises when one member provides nutritional or protective factors that is needed by the other. Parasitism, the host provides the parasitic microbe with nutrients and a habitat, and the parasite multiplication of the parasite usually harms the host to some extent. Now, antagonism, one of these non-symbiotic relationships, arises when members of the community compete with each other. One microbe can secrete chemical substances into the surrounding environment that inhibit or destroy other microbes. Antibiosis is a form of antagonism. This is the production of inhibitory compounds, such as antibiotics, by one organism that will destroy somebody else. Then synergism is that interactive relationship between two or more free-living organisms that benefit both but are not necessary for their individual survivor. Basically, they're better together than they are single. Now, what about biofilms? Well, really, this is the epitome of synergy. Biofilms are mixed communities of different kinds of microbes, and they exist together for the benefit of the entire community. Usually we have a pioneer colonizer that initially attaches to a surface, and we do need some sort of surface on which to actually start a biofilm. Other microbes then are attached to the pioneer or to the polymeric or protein substances produced by the colonizers, and then that attracts even more. And this attraction is known as quorum sensing. Cells are stimulated to release chemicals as the population grows to monitor its size and to bring in new organisms. So here we have a pioneer bacteria colonizing a surface, and it will excrete extracellular material that keeps them on the surface and it serves as attachment points for later colonizers. And quorum sensing chemicals, which are represented by our little red dots here, are released by the bacteria. But you'll notice now that in many, but maybe not all biofilms, other species will join and contribute to the extracellular matrix and or participate in that quorum sensing. And so here we see several different organisms now relying on each other in this biofilm. Mature biofilms may stop or minimize quorum sensing, and as biofilms serve as a constant source of bacteria that can escape and become platonic once again. And so these guys can go out and start another biofilm or again might cause damage somewhere else. So here's a good example of a biofilm actually on the surface of a tooth, and this is good old tooth plaque. Now, I talked a little bit about the normal microbiota. These are microbes that normally live in and on us and are part of our makeup. They live everywhere, actually, and they participate in commensal, parasitic, and synergistic relationships with the human host. So, pathogenic microbes have an optimum growth temperature in the what range? A what type of anaerobe can grow in the presence of oxygen but does not require it for its growth? An obligate what inhabit the Dead Sea? And in what type of relationship does one member benefit while the other is being harmed in some way? Now let's talk a little bit about population growth. Most of our bacteria, especially, reproduce by binary fission. They make a copy of their DNA and they split in half. So the parent cell enlarges, the chromosome, and I should say chromosome, is duplicated. The cell envelope pulls together in the center of the cell to form a septum and basically then separates into two genetically identical daughter cells. So simply making our copy of DNA, we form our septum, and eventually then the two cells separate. We can look at the growth of a microbe by looking at its generation time or doubling time. Generation time is the time required for a complete fission cycle. So for one cell to become two cells, or two cells to become four cells. In other words, the population is doubling. How long does that take for that doubling to occur? Each new fission cycle doubles the population. And as long as the environment remains favorable, the doubling effect can continue at a constant rate. The length of the generation time is a measure of the growth rate of the organism. Now, microbes grow exponentially. So the growth pattern of most microbes is going to be useful in expressing the population of microbes as exponents or logarithms. Plotting the data from a growing bacterial cell population, the number of cells is going to be plotted against the function of time. And this can be represented logarithmically, which will be a straight line, or by the pure raw numbers, and this will actually be a curve. So we can see if we start out with one cell and it undergoes binary fission, becomes 2, becomes 4, 8, 16, 32. The first generation, one cell became 2, the second generation, 2 became 4, etc. And we can actually express that exponentially. So the first generation is 2 to the first, second generation 2 to the second. What we'll see here in this graph is the log number or the actual numbers here in the red. And you'll notice that it's kind of slowly increasing, and then all of a sudden, whoop, up we go. If we actually plot that as a log of the number of cells, then it winds up being a nice straight line. What this is referred to is the growth curve, and this is a predictable pattern of growth within a population. And this can be applied to any population. And we can look at the number of microbes as a viable count technique. Traditionally, it's used to observe the population for the growth pattern. The total numbers of cells are counted over a given time period, and we'll talk about that a little later. This is a fundamental method of laboratory microbiology. Now, we can actually do plate counts. And in this, what we're going to do is we are going to take an inoculated sample and we're going to put 0.1 milliliter of that sample into 0.9 mils of fresh media. And we're going to then plate it, take a sample from the tube at every hour. And then we're going to count the number of colonies on the plate. So when we start here at 60 minutes and we took a sample, we got none or one colony forming units. Well, we know by mathematical equations, because of the logarithmic growth, that we have less than 500 cells then in this particular flask. But as we go further along and take samples, that exponential growth, you'll see that at 2 hours, now we have 2 colony forming units, and then 4 and 7 and 12, and it goes out here to 230. By doing the math, we can then figure out how many microbes started out here in our original flask. Who wants to count a plate of 230 microbes or colony forming units? And in fact, this becomes very difficult because oftentimes they start to overlap. But what we want is a plate somewhere between 25 colonies and 300. Depends on what book you read, whether it's 25 to 250 or 30 to 300. But probably what we want is a good plate somewhere around 100 colonies. And so either this 80 or the 135 plate would be really good to do the math to figure out the actual estimated number within the flask. So by doing the math at 135, we know that we started out with 675,000 individuals within the flask. Now let's look at a normal growth curve. First of all, we're going to have what's known as the lag phase, and this is a flat period on the graph where the population appears not to be growing. They are not reproducing. They are doing metabolism, however, getting ready for reproduction. And so newly inoculated cells require this lag period of adjustment, enlargement, and metabolic synthesis in order to get ready for binary fission. However, once we start to undergo binary fission, we then enter the exponential growth or growth log phase. This is the period during which the growth curve increases dramatically. And again, we usually plot this as a logarithmic curve so that it's a fairly straight line going up at about 45 degrees. This phase will continue as long as the cells have adequate nutrients and the environment is favorable. If, however, the population enters into a place where the nutrients are declining and waste products are beginning to build up, we enter the stationary phase. And this is when the population enters survival mode. The stationary phase actually flattens on the curve. The cells stop growing or grow more slowly, and for every new cell that's produced, a cell dies. If nothing changes in the environment, no new nutrients are added, and the waste products are not taken away, the microbes then will enter the death phase. So limiting factors intensify and the cells begin to die, and interestingly enough, they die at an exponential rate as well as growing. So the curve will dip downward, and again, producing it as a log growth curve, we actually will see this line dipping down at 45 degrees. So here's our normal growth curve. So again, that lag phase is that short period of time usually where the cells are acclimating to their new environment, they're getting busy and doing metabolism and getting ready to undergo binary fission. Once that happens, they then begin to grow exponentially until the nutrients begin to be used up and waste products begin to accumulate, and then we enter that stationary phase. At some point, if nothing changes, we then enter that death phase, and again, these cells will die off at an exponential rate. Now, a few cells may remain viable, but if this continues completely, all of the cells eventually will die. Antimicrobial agents can rapidly accelerate the death phase. So microbes in the exponential growth phase are more vulnerable to these agents than those that are in the stationary phase. Those in the stationary phase are already having to do things to survive the lower nutrients and the higher waste environment. So those that are in the growth log phase are actually more vulnerable to the antimicrobials. Growth patterns also can account for the stages of infection. Bacterial shedding usually occurs during the early and middle stages of infection and is more likely to spread to others during these early phases. Now, I could actually figure out a way to actually keep a colony or a population growing pretty much indefinitely, and this is known as a chemostat. This is a continuous culture system that admits a steady stream of nutrients into the media while removing waste products. So it maintains the culture in a biochemically active state, and so basically this growth population will be set in the growth phase forever, as long as, again, as the nutrients are being supplied and the dead cells and waste products are being siphoned off. Now, I can actually measure growth by looking at how many microorganisms are in there. And an indirect method of that is turbidity. Turbidity is simply how cloudy a substance is. And the more microbes that are growing, the more turbid a solution becomes or the media becomes. So a tube of clear nutrient solution becomes cloudy or turbid as the microbe numbers increase. I can also do direct or total cell counts. And one of these is simply the plate count, where we count the number of colonies on the plate and then do the mathematical equations to estimate the total population. Or we can actually directly count cells under the microscope. This uses a cytometer, which is calibrated to accept a tiny volume of sample spread over a pre-measured grid. And we can then actually count the number of bacteria within each grid and again apply a mathematical equation and get the approximate number within the total volume. So this is one way that we can use turbidity, and that's using a spectrophotometer. In a spectrophotometer, we have a light source and a plate that actually will measure the amount of light that passes through a sample. So if we have low turbidity, basically all of the light is transmitted and we'll have 100% of transmission. However, if we have a cloudy or turbid sample, only some of the light waves get through. Some are refracted by the individual organisms within the media, and so we get a lower percent of light transmitted. And again, based upon percentage of transmission, we can actually then do a mathematical equation and figure up some approximate numbers, but they're going to be a very, very wide guess. So we can say we have somewhere between 2 million and 8 million within a sample. It's not really very accurate. The direct microscopic count using this cytometer slide is we actually have a specialized grid that is divided into 25 large squares, and this is one of the large squares that actually have 16 smaller squares in each one. What we then will do is under high power, we will count the number of microbes within one of the large squares. So I can count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Then I'll go to the next large square, count how many is in that. After I've counted all 25 squares, I will take the median of that, and then again, applying mathematical equations, I can get a very close estimate of the actual numbers within the entire volume I started with, because we have a pre-measured volume that sits on this grid. We no longer do this by hand. We actually have machines that you simply put this cytometer slide in, and it does all the mechanics for you and the counting and the mathematics and spits out a number for you. But this was actually a way that I learned to count bacteria when I was in bacteriology eons ago. The culture counter is an electronic scanner that scans a culture as it passes through a tiny pipette. Each cell is detected and registered on an electronic sensor. And so it does count the total number of cells. But the flow cytometer is a much more accurate count because while it's similar to a culture counter, it will measure the cell size and differentiate between living and dead cells. So it will only count the living cells that were in the sample. So this is actually a flow cytometer, and individual cells will pass through this counting orifice, and in the flow cytometer there's a laser light that actually passes through here. If the cell is a living cell, it will be registered on the electronic detector. If it's not, it will not be detected, and so we get a very automated accurate count. And again, based upon the volume we put in, we can do the mathematics and go back to our original sample. Newer methods for analyzing population growth include polymerase chain reaction, or PCR. This quantifies the bacteria and other microbes in the environment and tissue samples without actually having to isolate and culture them. What we're going to do is count the number of pieces of DNA. And based upon knowing the bacterial's genome, we then can go, okay, so I had this many pieces of this particular piece of DNA, and then I can go back and say, then I now have that many bacteria that would have produced that many pieces of DNA. We can also use tests that measure metabolic activity and primarily measuring the amount of ATP that is being produced. However, this is primarily used in food and pharmaceutical industries and not in laboratory settings. It can be used for rapid quantification of microbes in certain environmental samples, however. So, true or false, the average generation time of most bacteria under optimum conditions is 24 to 48 hours. Can you place the 4 steps of the growth curve in their proper order, and do you know what each one represents? And finally, what is the machine that can differentiate between living and dead cells? So this is the last concept check, so see you in the next chapter.